Greetings. My name is Tony Fauci, and it is my great pleasure to provide this keynote address at the Global Vaccine and Immunization Research Forum 2021. I would like to thank the organizers for inviting me to present. I also thank you for all your good work, especially during these difficult times. As you can see from this title slide, I'm going to be speaking to you over the next few minutes on the subject of vaccinology in the context of pandemic preparedness, using the COVID-19 experience as the backdrop of this discussion. I've divided it up into four separate components. The first is to remind people of the commitment we had to what was called the decade of vaccines, in which much has been accomplished and there still is much left to do. The decade of the vaccines, which is 2011 to 2020, there has been progress towards goals for very elusive diseases with regard to vaccines, but progress has been made in both TB and malaria and in HIV. We've increased the clinical trial capacities, and we've embraced new vaccine technologies, novel genetic approaches, structure-based vaccine design, and a variety of new vaccine platforms. The goals of the decade of the vaccines are shown on this slide. Achieving a world free of polio, meet the vaccination coverage targets in every region, country, and community, exceed the Millennium Development Goal 4 target for reducing child mortality, and the meeting of global and regional elimination targets and the development and introduction of new and improved vaccine and vaccine technologies. So let's take a look at vaccinology in 2021. John Mascola, Barney Graham, and I wrote a few pieces in JAMA and in Nature Reviews Immunology in which we attempted to describe the technological advances that have been made in the latter part of the 20th and the first couple of decades of the 21st century, which have really become essential components of an adequate response to emerging viral diseases. And they're shown on these two slides. The first is the rapid genetic sequencing. What we did was we said we were going to apply new science, which is what I'm showing you on these slides, to an old discipline, which is the discipline of vaccinology. So besides the rapid genetic sequencing, which is very critical to all we do, there's the delineation of atomic level structures of viral proteins, the structure-based vaccine design, the cell sorting and sequence technologies, genetic knock-in technologies to develop animal models with human antibody genes, new vaccine platform technologies, mRNA, DNA, vector expression, nanoparticles, new adjuvants to improve immune responses to vaccine antigens, and reverse vaccinology. Now, what about pandemic preparedness and response in the context of these new technologies that represent vaccinology in 2021? Let's take a look at it from the concept of vaccinology and emerging infectious diseases, with vaccine development having two components to it. One is preparedness. Now, we at NIH have taken a three-part approach to vaccine development for emerging infectious diseases. We refer to it as our 21st century approach, and it involves the three components shown on this first slide. First is priority pathogen approach. By that, we mean to determine ahead of time those pathogens that most likely will emerge. And in fact, the WHO R&D blueprint has named a number of priority diseases that are shown on this slide. This has a role in how you address emerging infections, but we must remember that most of the infections that have turned out to have a global impact were ones that we had not anticipated. So although it is important to anticipate and prepare, it is likely we'll have to deal with some unknown virus like those that we have dealt with over the past couple of decades. The next is the improvement in vaccine platform technology. And as shown on this slide, there are a number of new technologies that have now really dominated our approach to COVID-19. For example, there's genetic immunization using RNA and DNA vaccines. We are well aware now of the successes, for example, of the mRNA vaccines. And there's the viral vector using things like adenovirus vector, which again, an important component of our armamentarium for COVID-19. And then again, recombinant protein, yet again, another important component, including nanoparticles with the viral proteins put on particles. And then VLPs, such as seen in a number of other vaccine attempts and accomplishments that we've done. And of course, the mentioning of a number of new and improved adjuvants. Then there's what we call the prototype pathogen approach. By the prototype pathogen approach means that you study pathogens of a particular family or group so that you build on prior experiences that you've had with similar pathogens in the same category. Because quite frankly, you can apply strategies and tools from one virus to inform vaccine design for related viruses, such as the basic virology, the assays for preclinical and clinical settings, animal models that would be interchangeable, antigenic targets, optimum platforms, potential immune correlates, and strategies for manufacturing. Some examples. We've known about human coronaviruses for decades. In fact, those shown in red are the human coronaviruses, and the four that are colors in yellow background are the common cold coronaviruses that infect us repetitively each year and for which we've studied for some time. Then there was the experience in 2002 with SARS, the original SARS that we now call SARS-CoV-1. And then in 2012, the Middle East Respiratory Syndrome, or MERS. Until now, we're dealing with the historic pandemic of SARS-CoV-2 causing COVID-19. So these are some of the examples of work that was done years ago from this 2008 paper in which we developed a DNA vaccine phase 1 study for SARS, which disappeared and negated the need to develop to its fullest a vaccine against SARS-CoV-1. And then with MERS, the same thing, but it was important because it was here that we applied the structure-based vaccine design to show the importance of the pre-fusion spike antigen of MERS-CoV-2, which held us in very good stead when we went ahead with the SARS-CoV-2 vaccine. I'll get back to that in a moment. Then there is the response to emerging infectious diseases using vaccine technology. So let's take a quick look at the incredible impact of COVID-19 globally. As of 2/17/2021, there have been over 110 million cases in the world and more than 2.4 million deaths. As I mentioned, the prototype pathogen preparedness enabled us to rapidly get a successful vaccine, particularly with mRNA and the experience that I mentioned to you that we had first with the SARS-CoV-1, then the MERS, and now using structure-based vaccine design to stabilize the spike protein in its pre-fusion conformation, which we had demonstrated was dramatically better in immunogenicity and protection than is the post-fusion confirmation. In the United States, we've taken a strategic approach to COVID-19 vaccine R&D. Namely, we've supported the development of and/or clinical trial of a number of vaccines that, for the most part, have gone under the same protocol design, the same clinical endpoints, except for one, the same data and safety monitoring board, and a variety of other components that were comparable among them. These are the vaccines shown, the 3 separate platforms and 6 separate companies, the mRNA, the adeno, and the recombinant protein. You're all aware of the history of these. The 2 mRNAs, which have already received EUA for 94 and 95% efficacy against symptomatic disease. The Janssen, which soon will have the data that was submitted to the FDA be presented to their VRBPAC or advisory committee. And then we have a number of other vaccines that are in the process of development, some soon to be getting an evaluation for an EUA. One thing to point out, that the speed with which we went from appreciating the new virus, in this case, SARS-CoV-2, on January 10th of 2020, to starting a phase 1 trial about 65 days later, a phase 3 trial, both with Moderna and with Pfizer, on July 27th, and now having both vaccines already being administered here in the United States and elsewhere. The fastest in the history of vaccinology, from the identification of a particular pathogen to the actual administration of a vaccine. These are just some New York Times articles showing about the efficacy of both the Moderna and the Pfizer product. As I mentioned, other candidates are right behind there, with Novavax in a UK study showing almost a 90% efficacy. In fact, it was also tested in South Africa, and although it only showed about a 49 to 50% efficacy against symptomatic COVID-19, it was quite good in cases of severe disease. The J&J announced a single-shot Janssen COVID-19 vaccine, which is, as I mentioned, currently being evaluated for an EUA. And then finally, the challenges for the next decade, which can be summarized in this article from Vaccine in July of 2020. Improving existing vaccines, developing novel vaccines for potentially vaccine-preventable diseases, and finally, and importantly, to ensuring the equitable access of vaccines to all people, all races, all classes throughout the world. Thank you.